The Big Three by Emma, Macy, Tassos, and Shane.
DNA replication is the process of producing two identical replicas of DNA from one original DNA molecule. DNA replication starts at the origin. At the origin, the helicase comes in and unwinds the DNA. That's why it starts to open up and look unwound. Then comes the DNA polymerase, building the new strand at 5' to 3' direction. This means it moves along the old template strand in 3' to 5' direction. When the DNA polymerase is building the new strand, the bases pair together with hydrogen bonds. The base adenine pairs to the base thymine, and the base cytosine pairs with the base guanine. The strand that is made is called the lagging strand because there are different sections that need to be paired together. These sections are called Okazaki fragments. Enzymes need to be placed down to pair them together. Primase prepares the segment for replication. Then, ligase comes and glues them together. And just like that, you have fully replicated DNA. Transcription is the production of messenger mRNA by the enzyme RNA polymerase and the processing of the resulting mRNA molecule. The first part of transcription is when the helicase unwinds the strand into an open-looking complex. Then, the RNA polymerase moves along the template strand, synthesizing an mRNA molecule. Once the strand is synthesized, it gets longer thanks to the new nucleotides. The RNA transcript is nearly identical to the coding strand of DNA. The RNA polymerase will keep transcribing until it gets signaled to stop. The process of ending transcription is called termination. Then it transferred out of nucleus into RNA.
Translation involves decoding a messenger RNA and using its information to build a polypeptide, or chain of amino acids. For most purposes, a polypeptide is basically just a protein, with the technical difference being that some large proteins are made up of several polypeptide chains. Each tRNA has an anticodon, a set of 3 nucleotides that binds to a matching mRNA codon through base pairing. The other end of the tRNA carries the amino acid that's specific by the codon. In translation, the mRNA is always read from 5' end towards the 3' end. There are 3 places on the ribosome where the tRNAs bind, the A, P, and E site. The A site accepts the incoming tRNA bond to an amino acid. The P sites hold a tRNA that carries a growing polypeptide. The E site is where a tRNA goes after it is empty, meaning that it is transferred to its polypeptide to another tRNA. tRNAs bind to the mRNAs inside of a protein and RNA structure called the ribosome. As tRNAs enter slots in the ribosome and bind to codons, their amino acids are linked to the growing polypeptide chain in a chemical reaction. The end result is a polypeptide which amino acid sequence mirrors the sequence of codons in an mRNA.
Important key terms. 5' to 3'. This is the direction DNA and RNA is read in. This refers to carbons on sugars, 5' being phosphate and 3' being hydroxyl. Adenine, this is a nucleobase. Adenine is one of 4 bases of nucleic acids. This is paired with the other acid called thymine. Amino acid. These are organic compounds that combine to make proteins. Anticodon. An anticodon is a sequence of 3 nucleotides forming genetic codes in tRNA responding to codons in mRNA. Antisense strand. This is a segment within double-stranded DNA that carries translatable codes 5' to 3' direction. Attachment sites is where the amino acids attach to the codons. Codon is a sequence of 3 nucleotides that together form genetic codes in DNA or RNA. Cytosine. This is one of 4 nucleic acid bases. This acid is paired with guanine. DNA. DNA stands for deoxyribonucleic acid. This is a main constituent of chromosomes. DNA is the carrier of genetic information. Exit site. This is the third tRNA ribosome, which is where the protein exits the polypeptide chain. Guanine. This is one of the 4 nucleic acid bases. This acid is paired with cytosine. Lagging strand. This is one of the strands of DNA found in replication. There is a slight delay before undergoing replication. Leading strand. This is one of the strands of DNA found in replication. This strand is read from 3' to 5'. Ocazoic fragments. These are short sequences of nucleotides and are linked to the ligase enzyme to create the lagging strand. Peptide bonding site. This is the second RNA ribosome, which is where protein is made. mRNA. This is messenger RNA, which is a single-strand RNA molecule, which is complementary to a DNA gene strand. rRNA is a ribosome RNA, which is a non-coding type of RNA that builds ribosomes, and protein synthesis occurs in the ribosomes. tRNA. This is the transfer RNA, which is an RNA molecule that decodes mRNA into protein. rRNA, this stands for ribonucleic acid, which is a polymeric molecule for coding, decoding, and expression genes. RNA codon chart. This is a chart with 20 amino acids and 64 anticodons that is used to build a polypeptide chain. Protein synthesis. This occurs in both transcription and translation. This is the process of cells making proteins. Ribosome. These link amino acids together in order of mRNA. RNA primer. This is a short single-stranded nucleic acid that initiates DNA. Semi-conservative. This is a process found in replication by denotating nucleic acid, in which one complete strand of each double helix is derived from the parent molecule. Sense strand. This is a strand of DNA with the same sequence of mRNA, which takes the antisense strand and eventually makes protein. Uracil. This is one of 4 nucleic acid bases. This acid is paired with thymine.